So now we're going to get into the actual process of mitosis and whatnot. Now, it's important to note when we're looking at things like replication, the basic structure of DNA. So we're going to quickly review that before moving forward. So remember that we have a sugar phosphate backbone on both ends, and then the bases in the middle, bound together by hydrogen bonds. Now let's get into the actual dividing process, where we look at mitosis and the purpose and process of it. There's two main reasons we'll go through mitosis: growth and repair. Instead of cells getting bigger, we just make more of them when we want to grow. And when cells get damaged beyond being able to fix them, we replace them by going through mitosis as well. And mitosis, because we're going through growth and repair as our reasons behind it, we're producing identical cells. So this is almost like a cloning process. So we're adding more cells to growing organisms because there's a limitation on how large cells can be. Cells are stuck being really teeny tiny because we need a larger surface area in comparison to the volume. Because if the volume gets to be too large, then it's really hard to go through diffusion, osmosis, and other things. And if cells are lost or damaged because something has happened, an injury of some sort, an illness of some sort, then we have to replace them with the same type of cell. And a lot of times cells just are too old or damaged just because time has gone by. Cells are exposed to the environment and UV light and other things that chip away at the integrity of the cell. And so when that point is reached where the damage or the age of the cell is impacting its ability to work, we undergo a process called apoptosis, or programmed cell death. So the cell is programmed to go through a process where it literally will self-destruct. This process is absolutely necessary because it's going to prevent us from developing things like cancer or randomly dying and potentially releasing things that are dangerous into the surrounding area. So it's a very controlled process to go through apoptosis. Now, eukaryotic chromosomes can be in several different configurations before and during and after mitosis. So depending on where we are in the cell cycle, chromosomes have a slightly different kind of look and a different kind of appearance. So before mitosis, the chromosomes replicate during the S phase of interphase. It's super important to remember that the chromosomes replicate before mitosis, right? You would try to write your paper and proofread it before you hit submit. You wouldn't submit a blank assignment. Now, the two chromosomes, once they've gone through and been replicated, are held together at a pinch point called the centromere. Now, I'm going to be talking a lot about different terms that are going to sound really similar. Chromosomes, chromatin, centromere, centrosomes. So you'll really want to pay attention to the language here. I'll try to kind of point it out and emphasize it, but you'll definitely want to keep your ears open from this point forward when it comes to vocabulary. So we have two identical chromosomes that are held together at a pinch point called the centromere. And until they're separated, so the entire time that they're held together at the centromere, we call them sister chromatids. This indicates that they are still held together and that they're identical to each other. So a better term would be twin sister chromatids, since they are identical twins. That DNA replication process made them into exact copies of each other. And so they're two copies of one chromosome. This will be important later on because this is what's going to allow the new daughter cells to have identical DNA to each other. Now, in order for the sisters to become a more manageable format, rather than just long, loose chains of nucleic acids, we go through a condensation process. So we condense down, get really densely packed, coiled together so that we can manage and move these chromosomes around a lot easier. Because we're going to do some moving and shaking. So let's look at the chromosome. So this is during any other part of the cell cycle except for mitosis, where we have a chromosome that's all spread out and long and uncoiled. And so we see this long linear link of DNA. We go through replication, and now we have two chromosomes that are 100% identical to each other. They are the same in every way possible, and they're held together at this point we call the centromere. Now, these guys are long and loose, and so it'd be really hard to move them around, which we'll have to move them in the near future. So we start to condense them down into tighter and tighter packed groupings. And so then we finally get the kind of famous chromosome shape, which is composed of 2 densely packed, condensed down sister chromatids. So let's go through an overview and then kind of go into the nitty-gritty of mitosis. We start with a parent cell. We've replicated that DNA in the parent cell during interphase. Then we can go into mitosis, where we condense those chromosomes down. We line the chromosomes up along the middle or the equator of the cell. We pull each of those sister chromatids in different directions so that one chromatid goes one way and the other chromatid goes the other way, pushing them to opposite sides of the cell, and then finally dividing that cell into 2 so that each new daughter cell has 1 chromosome from every single pair. So let's go ahead and look at this now in more detail. Sometimes people put the process of mitosis in stages called IPMAT, which doesn't really have a good ring to it. But let's go into it and kind of talk ourselves through this. Interphase is what's happening before mitosis, and this is where we replicate that DNA, and we have uncompacted, kind of loose and long chromosomes. So when we think of interphase, we're thinking of kind of the generic cell, just going around doing its daily cell activities. But then we get into mitosis with our first phase of mitosis, which is prophase. In prophase, the nuclear membrane is going to dissolve. We're going to have to move these chromosomes around, and as long as the membrane of the nucleus is there, it's in the way. So it starts to break down. The chromosomes condense down into those tightly packed chromosomes we are more familiar with, and the spindle forms on each end. So here's a centrosome made up of 2 centrioles, and here's another centrosome. They're going to move to opposite ends of the cell, and they're going to send out spindle fibers to grab onto the chromosomes. In metaphase, those spindle fibers are fully attached to the centromeres of each chromosome. And through a bit of a game of tug of war, we're going to line the chromosomes up at the equator in the middle, so that we have a centrosome here forming the south pole, a centrosome here forming the north pole, and their spindle fibers have fully attached to our chromosomes, pulling them in both directions until we finally line everybody up along the middle. In anaphase, we're playing tug of war, but we're basically snapping the rope in the middle. The sister chromatids are pulled in each direction so hard that each member of the pair goes one way or the other. So one sister chromatid from this blue chromosome goes towards the north pole here, and the sister chromatid for the blue one down here goes to the south. So we're pulling these chromosomes to opposite sides, and our cell is starting to elongate. Then we get into telophase and cytokinesis. Again, cytokinesis is not necessary for every single cell. There are some times where we don't go through this, although for the most part, telophase and cytokinesis kind of go together. In this case, we're kind of reversing what we saw in prophase. That nuclear membrane is going to come back around each set of chromosomes in each new daughter cell. The chromosomes decondense and become that just unfamiliar, no real pattern, no real design look that they had before. And we start to see the cell pinch into 2. We're also dividing up all the other components. So things like the cytoplasm and organelles are going to get divided up so that each new cell has the cytoplasm and organelles it needs to continue. And so we end up with 2 daughter cells that are 100% identical to each other and identical to the original parent cell.